Hello, everyone. Thank you for joining me today. From its inception, GemStone was designed for automation. As cytometrists were able to add more and more measurements to their analyses, we realized early on that the ability to automate high-dimensional data analysis would be necessary to provide efficient and objective results. Today, we're going to discuss the features that help to make this automation possible and unique. Previous webinars have covered a general overview of GemStone and probability state modeling, building an analysis model, as well as cleanup strategies using TRICOM to enumerate unknowns and the Sensei dimension reduction algorithm. For those of you new to GemStone, it is a comprehensive approach to high-dimensional flow analyses, incorporating a variety of features in one automatable program. The engine that runs GemStone is called probability state modeling. To the unfamiliar or for review, I refer you to this paper, as well as our YouTube channel, where you can find previous webinars and videos. You can also check our website for publications here. So once an analysis model is built, the workflow for automation is quite simple. Upon opening the program, you either create a new project or open an existing project. You then add your sample files, program the batch, and run the auto analysis. You can take a break and get some coffee while the program performs the analysis, saves the analyzed files, saves the reports and graphics, populates, saves, and exports the database. You can then review the reports, check for alerts, and continue with more in-depth statistical analysis if needed. All of the results are organized in folders for you. Reports can be printed out as well as saved if desired. So today we're going to look at these steps a little more closely, starting with opening or creating a project, then setting up the batch, running it, and finally taking a look at the results and where to find them. We will also talk a little bit about the database since it is part of the batch system. So I'm switching to a live program now. I'm going to end my slideshow. I have a version of GemStone open. And the first thing we're going to look at is opening a project. If you have been using the program, it remembers the last project that was open, and it will show you in the drop-down window here at the top left. I already have created a project for this webinar, which you can see in the window. A project in GemStone can be thought of as a full experiment, which includes data files, templates, settings, and results. When you build a model based on a specific panel, you will most likely create a new project. Thus, the project can function as a template for a new project using the same panel or an edited version of that panel. The saved project will save the batch system and the settings for the batch, so when you open it, you are ready to go. So we're going to look at how to set up the batch as we proceed. The first thing we want to do is add sample files. So we're going to click the Add button. And this brings up a navigation window that opens automatically to the project file folder. You can see at the top that I have the name and the FCS files. If you need to, you can navigate away from this window to whatever folder that your data files are saved in. I'm going to multiple select. I'm going to open my original and then multiple select my donor files. I have 5 separate donor files, and I'm going to click open. So these will automatically be added to the batch list. And by default, the program adds the data files to level 1, which you can see here on the left. We're going to talk more about these levels shortly. And then to set up the batch, you must be in develop mode, which is here. There's a little click box. So we're going to check that click box to activate the controls. When I check this box, the view changes to bring up a new tab, which is the develop tab here at the top. And it also exposes the batch commands, which are down here at the bottom. I'm going to open these up a little bit more so we can see them all. Now, the purpose of these different modes are so that you can run the program in compliance mode using user permissions, so effectively you can lock down the program if need be, or if you want to avoid making inadvertent changes to anything. So first, we're going to do a little bit of a tour to find all of our tools. I'm coming back to the Home tab. In GemStone, there are generally several ways to do things. On the main toolbar of the Home tab, you're going to see some controls here. So at the top, we have our add or delete data files from the batch list. We have a tool that lets us start at a particular point in that batch list. We can run one file in the batch that's highlighted. We can run the next. We can do an auto advance, which goes through the whole batch list, and then we can stop the processing at any point if we want to end the analysis. We have a tool to open an analysis document and a tool to save the analysis document. If I right-click on the batch windows, I see some of the same tools. I have a start tool. I also can clear the flags. If I've already done a previous analysis, I can remove one file or remove all files. And then I have some controls that relate to the database, which we're going to talk about a little bit more later. We also have a set of batch list controls. If I highlight this first file in the batch list, it activates these. We also have another tool to add or remove the data files. And then we have an indent and an outdent tool. If I highlight the file and then indent, it automatically switches that to a higher level. I can go back and outdent that file to the lower level. And then I can use my arrow tools to move a file down in the batch list or back up to any position that I want those files. So if I have an order in which I want my files analyzed, I have control over that. And then again, I can set my starting place here with this little tools. Next set of commands are what enable a user to determine what actions are performed on the data file and allows for this fully automated analysis. The levels actually allow for different actions to be performed for different types of related data files. So for example, this particular example has a 2-level workflow. It's a 33-color SciTech Aurora data file that I have 5 separate donors, and these are PBMCs. So the first analysis here, which takes place on level 1, uses a cleanup model, which cleans the file, removes the dead events, aggregates, and debris. That cleaned file is then exported to level 2 and appended with a suffix. I'm going to double-click on this first file just to demonstrate what happens. What's happening now is the program is opening that cleaned template. It is reading in the data file. It's performing the analysis while cleaning up that file. It's saving, it's creating a report and saving the report. And you can see here in the batch list that it has exported just the cleaned events from that file, created a new FCS file, and then appended it with a suffix so that I know that that is the cleaned file. It's also saved that document into the FCS files folder. So level 2 has been set up to perform the analysis using an analysis template. It also calculates a sensei, it creates graphics, it saves the analyzed sample file, populates an analysis database, and exports the database in a tab delimited form. So it's important to note here that the levels and the commands are user-defined and can be changed and programmed to suit your needs. Some analyses will only require one level, while others may have 4 or 5, depending on the goal of the analysis and the types of data files being used. So even if you're only using one level, this is still the means to automate the analysis. Some examples of the use of different levels include different levels for patient files and control files, different levels for different tubes or panels for patient data or experimental data, different levels for normal versus abnormal groups, or whatever else you can imagine or need to do in your analysis. So these level names are user-defined. You can type directly into these boxes, and I just decided for a simple title here, which is this is my analysis level, and my description is basically analysis. The controls here, I'm going to just start with level 2 and explain the different way that this batch is set up. So we do want, in this particular analysis, we do want to open a template file. I could also have the option to reset or not open a file at all. And then we come to the edit box. All of these edit boxes are active. When I want to add a template to be used in the analysis, I usually, for the location option, use the specified target file. I also have other options. It brings the target path to that model template folder, which is in the batch system and the batch folders. The target file is my 33 color panel. That window is active in a navigation window, bringing me right to the model templates. So I can pick the template that I want for that level. And then I click OK. This compact file tool is used generally for CyTOF and Helios data after the cleaning process. It helps get rid of any unneeded segments in that data file, making it just a little bit smaller. The read file should be on because I want to read in a data file. And then this particular panel is a 1-tube panel. I also have the options for doing a first tube level and then any number of levels for subsequent tubes. So if you have a panel that is 4 tubes or 5 tubes, you can do a different level for each one of those tubes, as long as they're related data files. Coming to the analysis tools, we have a normalized data, which is not exactly ready for prime time, but if anybody wants to collaborate on that, please let us know. I have an auto-analyzed data, which is what I do want to do here. I'm going to skip enrich and classify for now because that is covered in the user guide, and it's a little bit out of the scope of this presentation. If you have a sensei in your template, you want to click this on and it will automatically calculate that sensei for you. I can opt to print out my report. You can set up a printer where that report will automatically go to. And then I come to some tools for finishing up here that include my saving the document. So this is to actually save the analyzed data file. And you can choose the folder in which that gets saved into. So this depends on your lab preferences and your LIS system. So you can either choose a location option here, which you can bring it back to wherever that data file is located to, or it will automatically go into the fitted models folder here in the project library batch folder. And then I have my fitted models. Let's click that. I can save my PDFs. So this is for saving the PDF reports. Again, if I click on my edit box, it brings me right to my PDF reports, and I can choose how I want those to be named. I can automatically save my graphics. We have a separate folder for the graphics, which is set up for you in that batch. And then I can save my database, and this will also bring up a box that comes to the database where you can name the database however you want it to be named. Usually we use the specified target file to save that database in your naming scheme. And I'm going to say OK. A couple other options for that batch would be to synthesize data. If you want to have a known data file, this is an interesting option to actually synthesize a known file, which you can then use for further analysis, looking at different algorithms or whatnot. And then export and merge the FCS file. I'm coming back to level 1. And most of the differences in this level 1 between the level 1 and the level 2 is that I have a different template that was open. So this is using the cleanup model for the template instead of that analysis model. I don't have a sensei, so I don't have that checked. I haven't, in this particular analysis, I haven't saved anything because I wanted it to be fast. But what I did have is I had the batch export that data file, that cleaned file, onto level 2. And the tools here look a little bit different because I can choose the path. Automatically, it will come up in this exported folder in the FCS file folder. I can choose to have that go to a different folder if I wish. And then this suffix is user defined, so I could have that be anything I want. For simplicity, I just choose cleaned. You can choose whatever. Exported is something some people opt for. I wanted it to go to level 2. It can go to any level that you wish. And then my event filter chooses the events that I want to be exported. In this case, I'm exporting my cleaned events, which is coming from a certain cell type, and this would be cell type 2 in the list. There is another option here for merging files. If I have several different data files that are the same panel, I can merge them. I can start my merge on one level, add to the merge on however many levels I need to use, and then finish the merge on a separate level, which would then export a merged file. Okay. So it's important just to note a couple things about these batch levels. So when you click on a file and you indent it, it does create a new level. So I went up to level 3, it creates a new level. So this is one way to create new levels. All of these batch tools are then ready to be programmed, and they're active. So you can go ahead and type in a new name, you can add a description, and then you can start adding another template, just programming it down using all of these batch command tools. There are no limits to the number of levels that can be created. And each level is pretty much independent of the other level unless you designate otherwise, such as for first tube and subsequent tube merging files, etc. So the Auto Advance button will run through all of the levels in that batch file. So if you have hundreds of data files, if you click the Auto Advance, it's going to go through all of those on every level that you have programmed. Come back here. You can also delete this level. And I can't do it right now because I have files in there. So after you make any changes to your batch system, if you've done some extra programming or changed anything in the batch commands, you absolutely want to save your project. So when from the Develop tab, you can click this. You can also find this from the File tab. You want to save my project. It's going to ask you if you want to save the model template. If you've made any changes to the model template, you can say yes. Otherwise, you can say no. That won't hurt if you say no. But just make sure that you save the batch because it saves the programming and it also saves the database. So now that you have all of your programming done, you want to run your batch. Down here on the bottom left, there are some batch tools. One of the handiest tools, I think, is this little button right next to the batch title that brings up a pop-up window that has a list of all of the files in the batch. So if you need to scroll through and find if you have many data files in that batch list, this is a good way to find the one that you want without much trouble. You also have go to the first item, previous, you can do a run, the current item, you can click next, run the next item, again, the last item. This is auto advance, and then you can stop doing analysis altogether. The other thing that you can do is double click on a previously analyzed file and then bring up the analyzed document. So that would be one way to open an analyzed document. You can also use the tools up here. You can designate a starting place in any number of these locations. You can clear the flags. Again, if you've done an analysis previously, all of these will have flags. And this is actually a way to tell you where you are in that progression of analysis. If you have done just double click and redo that exportation to show you that flag again. So after I've done an analysis, you have this little check mark. So if you've been processing a great number of files and you take a break and come back, you'll see that check mark and you can tell where the progress is in your batch system. All the files are run in batch order, and then these analyzed files are flagged. You can clear that flag if you want, etc. So once everything's set up, all you do here is run auto and you're ready to go. If you want to set this up in compliance mode, you need to close the program, set up compliance, set up user groups, and then reopen the program in that compliance mode. And you'll come back to where you were here with just ready to go in your batch system with your batch controls active. So once you get your batch run, where do you find your results? In the installation folder for GemStone, there is a number of folders. Here's the application folder. One of the important folders here is this project library folder. If I open that, I can see my projects that I have listed, that I created, and I can look in any of these projects to find my results. So if I'm looking at this one, this is the one that I just created. I have a series of folders. This is the batch system controls that just sort of automatically get saved. This is where I find my databases. This is where the FCS files and that exported file will go to. My fitted models is here. I don't have any there right now because I didn't save any of the cleaned models. I also have a folder for graphics, my model templates, and then the PDF reports would be found here as well. Come back here and open that one. So I have a series of PDF reports, and my fitted models would be here as well if I went through that whole batch. So let's just switch gears a little bit now because I want to talk about the batch, about the database. There is an extensive database in GemStone. If you come to the Options tab, this is what the batch looks like here when I went through the whole thing and did all the batch processing. I'm going to double click on this information icon. This will bring up a window which is part of the information system. After you run one analysis, you will have all the information available, all the statistics that have been calculated for that particular data file that are now in the information system. All of these statistics, all of this information is available to be put into a database. So each one of these arrows, you can look behind and see what you have. There's equations, all my equations are here. So this is just a sort of a preview of what you have that's available to go in the database for planning purposes. If I come here to my fields icon, this is the actual database. On the right, you can see the database has been populated with. And on the left, you'll see what is available to go into the database. So if I want to add something, I can come here and I can highlight something and then add it. Whatever I add is added to the bottom of the list. If I want to insert something, I can highlight something and I can insert whatever it is I'm inserting. I could, just for an example, I'll just insert AutoComp. Not that you'd want to do that, but that's you can add something anywhere in this list that you want. You can also move things up or move it down or delete it altogether if you want to delete an entry. This edit field system can also be sorted. You can sort by cell type, and you can sort by percents only, counts only, or intensities only, which makes it a little bit easier to find something. There's a lot of information available here. Currently, this is set up with 104 items, but I could easily go to hundreds of items for my database. If I right-click, I can also find that edit fields box, the same view of that edit fields box. I can clear the results if I want to start over, and I can export the database from here as well and copy. So it's really important and helpful to spend a little bit of time to figure out what it is you want to put in the database. And once that's set up, again, that can be used over and over again. It's part of your batch system. On the Develop tab, the other thing that's important to know is that we have an Equations button. And this is a place to build equations. These equations can be turned into alerts, and those are available to be put in the database as well. You can do any kind of equation that you can think of or any kind of alert if you wanted. Here's an example of CD3 events. So I've added 4 different cell types to get my total CD3 events. And I could do some kind of an equation if I have, I want to look at CD8 or CD4 ratios. I can alert that if something's too high or too low. So saving a project, once you get everything set up, I mentioned before that you want to save the project. This saves all of the program that you've done. When you reuse the project, you can do a couple of different things. You can add data files directly to this batch list if you want to just reuse the whole thing. Keep in mind that the folders in that project library in the project are already populated with some previous results. You can either change the place where the results go to. If you want everything to go into those folders, that's fine too. That's user defined. But that's one way to reuse it. The other way to reuse a project is to click the New button, and you can create a new project based on a previous project. It will be appended with the date that you created that new project on. If I click OK, it basically kind of wipes the slate clean as far as results and data files, but all of the batch is set up and the database will be set up as well in part of that new project. If you had a completely new panel, you would just click and create a new project based on none or based on something similar, but I want to cancel that out. So I'm just going to switch back to my slideshow now, and just in summary, GemStone was really designed from the inception for automation. The batch system is a powerful tool for automating your analysis that is easy to use, it's customizable, it's flexible. During an auto analysis or auto advance, the program performs the analysis, calculates all the statistics, it populates the database, exports the database, creates and saves all the graphics, creates and saves the reports, calculates and saves a Sensei graph and a summary map, saves the analyzed data file. You can print out a report if desired. You can export an FCS file if desired, and more. So advanced options include multiple tube analysis, merging files, synthesizing files, multiple level analysis, and compliance mode. Projects save all of the setup and results that were generated and can be reused. And really importantly, projects can be shared with collaborators. So if you have multi-site studies that you're doing or multi-users that are trying to use the same panel, these projects can be shared and all of that program can be copied from one place to another. So I thank you for your attention. I thank you for joining me. I also thank SciTech for the Aurora data. You can contact us and see more information on our website. Always, I'm available if you want to email me if you have any questions. And I think we do have a few minutes left. If anybody has any questions, I'd be happy to answer them.